CRISPR gene editing, adapted from a naturally occurring genome editing system in bacteria, is a revolutionary technology, which allows genetic material to be added, removed, or altered at particular locations in the genome, and has revolutionized our ability to target disease-linked genetic mutations. The technology can target both DNA and RNA using different enzymes, Cas9 and Cas12 for DNA, and Cas13 for RNA. CRISPR germline editing therapies, or CGETs, have incredible potential to eradicate hereditary disorders, but hysteria ensued when a rogue Chinese scientist used the technology to edit the CCR5 HIV resistance gene in human embryos, triggering a funding moratorium by the NIH. In an open letter in Nature on March 13, 2019, a number of major players in CRISPR's development, including Emmanuel Charpentier, Eric Lander, and Feng Zhang, along with colleagues from 7 different countries, called for a total ban on human germline editing for at least 5 years until an international framework has been agreed upon. Notably, one of the key inventors of the technology, Professor Jennifer Doudna, has not signed this extremist document, saying that she does not advocate a possibly indefinite moratorium on the technology. Instead of educating the public about the costs and benefits of this amazing technology, researchers have decided to allow blind fear and emotionalism slow the progress of extremely important science able to eradicate disease and enhance human genetic fitness. This technology is not really awaiting any further advancements but can cure disease today. A recent article in Nature on a new CRISPR-based technique, called prime editing, pointed out that roughly 2/3 of known human genetic variants associated with diseases are single-point gene mutations. There are around 75,000 different mutations that can cause disease, and prime editing has the potential to fix 89% of them. Is that not impressive enough for you? In fact, these scientists are possibly understating the capabilities of CRISPR, because many gene mutations are additive, meaning that we don't necessarily need to fix every gene involved in a polygenic trait like intelligence to produce a positive effect. A recent study identified 1,016 genes associated with cognitive ability, but this still only explained a small proportion of the total variance. Stephen Su has suggested that as many as 10,000 genes are likely involved and that their additive effects explain the bell curve of IQ. It's important to note that our understanding of genetics is just beginning and new genes producing complex traits are being discovered every day. The irrational fear of gene editing technology derives from faulty assumptions made about heredity and human biology. It's quite comical to note that human reproductive decisions are the least regulated, while also being some of the most reckless. Few people think about the possibility of passing on inferior or diseased genes to their children, not to mention providing for them. The noted population geneticist Michael Lynch in the journal Genetics wrote that, quote, "What is exceptional about humans is the recent detachment from the challenges of the natural environment and the ability to modify phenotypic traits in ways that mitigate the fitness effects of mutations, e.g., precision and personalized medicine. This results in a relaxation of selection against mildly deleterious mutations, including those magnifying the mutation rate itself. The long-term consequence of such effects is an expected genetic deterioration in the baseline human condition, potentially measurable on the timescale of a few generations in westernized societies, and because the brain is a particularly large mutational target, this is of particular concern. Ultimately, the price will have to be covered by further investment in various forms of medical intervention." End quote. Lynch is referring to Kimura's neutral theory of molecular evolution, which is made particularly evident by genetic diseases associated with inbreeding. It turns out that evolution is more like a treadmill than a race. Nature is a very cruel force and it requires constant selective pressure to maintain fitness. When we don't intervene, nature gets to play its heartless games with our genome. Why is that a better default position? Lynch goes on to point out the human germline mutation rate is roughly 100 new mutations per person per generation, while the somatic mutation rate is roughly 3 new mutations per cell division. He estimates human fitness is declining 1 to 5% per generation. In other words, we are incredibly afraid of making even small edits to our genome, while nature is making edits all the time, and most of them are not beneficial. Furthermore, it's quite ignorant to think that a cheap technology like CRISPR will be easy to regulate, so bans will likely just force it underground. In fact, governments themselves are the most likely to use this technology for nefarious purposes. The idea that governments will keep this technology from being abused is laughable. If we ban this technology, the medical-industrial complex wins, and humanity loses. It's time for humanity to leave childhood behind and take responsibility for its own destiny. Regardless of whether CRISPR is allowed in the near future, cheap gene sequencing is already offering genetic screening for those having children. This may eventually lead to the selection of particular embryos or even specific gametes. I will leave you with a quotation from philosopher David Stove from his work, On Enlightenment. Quote, "Equality, externalism, and educationism are the basis, now even more than ever, of all government policy in the free countries, as they have been all along, and avowedly in the communist ones. Still, it is doubtful whether this farce can be protracted much longer. For we now have not only extensive knowledge of human genetics but a considerable capacity to engineer genetic change. For these reasons it is probable that the marriage between contraception and eugenics, which took place early in the 20th century but suffered a setback around 1940, will soon be resumed, this time with massive finality.